I'm Javairia Haq, and I've done prompt 1A for exam 3. DNA stands for deoxyribonucleic acid. It has a double helix structure and is a type of nucleic acid. It contains our genetic code. DNA's monomer is a nucleotide. A nucleotide is made up of 3 components: a 5-carbon sugar called deoxyribose, a nitrogenous base, and a phosphate group. In DNA, the sugar is called deoxyribose. For RNA, the sugar is called ribose. The carbons in the sugar are numbered clockwise. The second carbon distinguishes DNA from RNA. In deoxyribose, the 2' carbon has an H group attached, while ribose has a hydroxyl group. The 1' carbon attaches to the nitrogenous base, and the 3' carbon bonds to the next 5' phosphate of the other nucleotide. The 5' carbon is attached to the phosphate group. The sugar and phosphate group alternate to create the phosphate backbone. Phosphodiester bonds connect the sugar-phosphate backbone as the 5' phosphate connects to the 3' carbon of the nucleotide. The 5' end of DNA has a phosphate group, and the 3' end has a hydroxyl group. Because of this, DNA runs in the 5' to 3' direction, and each strand is anti-parallel to each other, meaning it's opposite of each other. Now, if we look at the inside of DNA, there are 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. The structures of these nitrogenous bases are shown here as displayed on medicinenet.com. Complementary base pairing between 2 strands of nucleotides allows for constant helix width. Cytosine pairs with guanine using 3 hydrogen bonds, and adenine pairs with thymine using 2 hydrogen bonds. Hydrogen bonds are relatively weak, and they are not the main force holding the 2 strands together. So, if we think about DNA as a ladder, the nitrogenous bases on the inside are hydrophobic, meaning they don't like water, and the phosphate backbone is hydrophilic, meaning it likes water. Due to base stacking, DNA will twist into the double helix structure to reduce the exposure of water to the hydrophobic bases. The hydrophobic interactions are the main force in stabilizing the double helix. In DNA, the major grooves occur where the backbones are far apart, and the minor grooves occur where they are close together. Normal DNA is right-handed, meaning if we look at the backbone closest to us and follow it up, it goes to the right. However, there are examples of left-handed DNA in media. If we look at the closest backbone and we follow it up, it goes up and towards the left.